Welcome to This Explainer. Okay, let's jump right in, because today we're uncovering a genuinely shocking biological twist in the progression of ALS. Now, we've spent decades trying to unravel exactly how this devastating disease systematically destroys the nervous system. But we are looking at some groundbreaking research published in Nature Communications that frames ALS not just as a mechanical failing of the body, but as a literal biological murder mystery playing out right inside the spinal cord.
Normally, we naturally assume the disease itself just slowly starves or kills off these cells, right? It's an intuitive way to think about neurodegeneration. But what if the brain's own immune system is the true executioner? What if the very cells tasked with protecting your neural circuitry are actually the ones pulling the plug?
Section 1. The motor neuron mystery.
So we all know ALS leads to the progressive loss of the vital motor neurons that control our muscle movements. That's what causes the terrible progression of weakness and eventual paralysis. But the actual mechanics of how these neurons die, well, that's harbored a pretty dark secret. Historically, we've focused heavily on the survival of the motor neurons themselves, but as you'll see, the dysfunction actually involves the neighbors.
Section 2. Microglia, the prime suspect.
Enter a type of cell called microglia. Now, what's wild here is that these are supposed to be the good guys. They're the central nervous system's tissue-specific macrophages. Basically, think of them as the protective cleanup crew. Their normal day job is just to clear out dead cells and keep the brain and spinal cord safe from infection.
But deep genetic analyses show a completely different, almost terrifying picture in ALS. These protective microglia become highly activated. They multiply wildly, and they absolutely swarm the vulnerable motor neurons in the spinal cord. It's like a mob creating a massively inflamed environment.
Section 3. The receptors of doom.
If they're swarming the scene of the crime, we have to ask, what weapon are they using? Researchers discovered that ALS microglia have massively elevated levels of something called TAM receptors, specifically two proteins known as Axl and Mer. Now, normally, they use these receptors to detect and eat cells that are already dead. But in ALS patients and mouse models, researchers found a modest increase in Mer and an absolutely staggering 16-fold increase in Axl on these microglia.
To prove these exact receptors were the weapon, researchers did something brilliant. They genetically removed the Axl and Mer receptors from the microglia of ALS mice, essentially disarming them entirely, and then compared them to standard ALS mice.
The verdict was incredibly surprising. Disarming the microglia did cause mice to show initial symptoms about 30 days earlier, due to some early inflammation. But here is the crazy part. They actually survived roughly 3 weeks longer overall. By the late stages of the disease, the mice without those TAM receptors had retained 3 times as many motor neurons in their spinal cords, and the connections out in their limbs were dramatically preserved.
Section 4. The Eat Me sickle.
Which brings us to the core focus of our explainer today. If removing these microglial receptors saves the neurons and extends lifespan, it implies the microglia were actively killing them. But we just established they're the cleanup crew. Why on earth would they attack and eat living motor neurons?
Here's the step-by-step of this biochemical execution. Sick, stressed motor neurons in ALS suffer from calcium overload. Because of this stress, they accidentally flip a lipid called PTDS to the outside of their cell membrane. Now, PTDS is a potent eat-me signal, normally only shown by dead cells. But these neurons are still alive. Once that signal is exposed, a protein called Gas6 binds to it like a homing beacon. Then, those hyperactivated TAM receptors we talked about detect the beacon. Tricked by this false flag, the microglia swoop in and literally devour the still-living neuron.
Section 5. Caught in the act.
We don't even have to just rely on biochemical theory here. Researchers got visual proof of this microscopic crime. We can actually see normal, wild-type microglia caught red-handed. Inside their cellular stomachs, you can clearly see them actively digesting motor neuron material. But the knockout microglia, the ones genetically altered to not have TAM receptors, they leave the neurons completely alone.
And the hard data backs this up perfectly. When researchers quantified this, they found a nearly 10-fold reduction in motor neuron material getting eaten when the AJ-Axl and Mer receptors were removed. The predation practically stops in its tracks.
Section 6. Rethinking ALS progression. So, what's the big takeaway from all of this? Microglial predation in ALS is, tragically, just a huge biological misunderstanding. The immune system genuinely thinks it's doing its job. It sees that eat me signal and tries to clear out what it assumes is a dead cell.
But in the process, it prematurely destroys the live, functioning circuits that keep the body moving. It's a protective mechanism gone horribly wrong. As the researchers so powerfully state, this active killing by our own immune cells actively accelerates the fatal progression of the disease. They aren't just sweeping up the aftermath, they're actually driving the decline.
And that leaves us with a deeply provocative dilemma. We could theoretically develop drugs to block these receptors, stopping the immune system from pulling the trigger, right? But those motor neurons would still be sick. They'd still be suffering from the underlying disease that made them raise that white flag to begin with. We can stop the executioner, but how do we cure the sickness? It's an incredible question, and one that's definitely going to drive the next generation of ALS research. Thanks for joining me on this explainer, and keep questioning the science.